Proteins are vital to the function of cells. In order for each organism to meet its needs, specific proteins are constructed based on its DNA, which contains the instructions for building them. First, the codons in DNA, which are clusters of 3 nitrogen bases, are copied onto an mRNA strand in a process called transcription. This mRNA leaves the nucleus and travels to ribosomes in the cytoplasm. Here, the mRNA is met by a strand of tRNA that brings amino acids to bond to corresponding codons. The process in which the amino acids assemble into a protein and leave the ribosome is called translation. Though both proteins and nucleic acids, such as DNA, are made with the same instructions, the former is composed of amino acids while the latter is made of nucleotides. It is this difference in composition that allows proteins to repair body tissue and regulate internal processes, functions out of reach for nucleic acids. Another key difference is that the body possesses DNA on its own, while amino acids needed to construct proteins must be gained either from food, in the case of heterotrophs, or from absorbed nutrients, in the case of autotrophs. Proteins are needed for life, and it is important to understand how they are formed, for the central dogma of biology is the idea that information from DNA is used to synthesize proteins. It is because of this that genetic mutations can be so devastating to organisms. Of the 2 main types, frameshift mutations have the greater potential for damage. Because a nitrogen base is either added or deleted, every codon after it will be altered, which can completely ruin a sequence. Substitution mutations are less consequential. This is when only one base is changed, and there are 3 outcomes. If the altered codon happens to code for the same amino acid as the original, then it is a silent mutation, and there are no effects. But if it codes for a different amino acid, then it is a missense mutation, and part of the protein will be incorrect. Depending on the importance of said protein, the organism may not function properly. Finally, if it is a nonsense mutation, the altered codon codes for a stop, and the sequence will be prematurely terminated.